Hi, my name is Tatiana Bahia. I am a sophomore at the University of North Florida. Over the summer, I interned in Dr. Frank Smith's lab. I was assigned the gene goosecoid, which is a transcription factor. It regulates the development of the foregut in other animals. We wondered whether goosecoid regulated the foregut in tardigrades. Tardigrades are microscopic invertebrates. They are segmented and have 8 legs. Tardigrades exhibit several similarities to model systems, such as roundworm and fruit fly, but little is known about the development of the tardigrade gut. We started our research by doing a conserved domain search. You can see that down here. We used 4 different species in our conserved domain search. We used 2 tardigrade species, Ramazodius and Hypsibius exemplaris, which is the tardigrade species that we study. And we also did fruit fly and mouse. From there, we were able to make a phylogenetic tree, which you can see here. The 2 tardigrade species are highlighted in red, and the outgroup is in black. From there, we wanted to see where goosecoid was expressed in tardigrade embryos. And to investigate this, we did an in situ hybridization experiment. We used embryos in 3 stages: 24 hours post-laying, 35 hours post-laying, and 45 hours post-laying. We then imaged the specimens using 2 different imaging techniques: differential interference contrast and confocal imaging. At 24 hours post-laying, goosecoid expression was the weakest. Expression was weak and not very highly concentrated. Goosecoid was the most highly expressed and highly concentrated at the 35 hours post-laying stage. You can see a dark staining where the foregut is. Expression was the most concentrated in the foregut at the 35 hours post-laying embryo, which you can also see on the confocal image. In the 45 hours post-laying stage, goosecoid was highly expressed, but less concentrated than at the 35 hours post-laying stage. It was more concentrated than at the 24 hours post-laying stage, but it was exhibited more broadly than at the 35 hours post-laying stage, which you can see here how there's a dark staining where the foregut is, but it's also darker throughout the embryo compared to 35 hours post-laying. So to conclude, we found that tardigrades retain an ortholog of goosecoid, and that goosecoid is most likely regulating foregut development in tardigrades. We found that goosecoid was the most weakly expressed at 24 hours post-laying, and it was more strongly expressed at 35 and 45 hours post-laying. Some future directions we plan to take this research in include using RNA interference to investigate the function of goosecoid in tardigrades, as well as comparing goosecoid's expression to other gut patterning genes, and using goosecoid to establish a more comprehensive model of the tardigrade gut, because little is known about the tardigrade gut. Lastly, I want to thank the NSF for funding this research. It has been an incredible opportunity, and I'm forever grateful.